Hello everyone. First to say a big thank you to the CPM team for organising this event. You've brought a wonderful group of people together, and I'm really happy to start off this discussion. So I'm actually going to give you a little bit of an overview to try and set the scene a little bit, give you some backgrounds necessary to understand all of this, and also kind of just highlight what the next speakers are going to talk about before passing on to them to give you some more details. So I'm going to start off by introducing you to Charlie. So this is Charlie shown here on the right with his dad, James. Charlie is a 12-year-old. He has a global developmental delay and intellectual disability. Those are both two very broad terms that are often used to describe developmental conditions in children, and they're often quite confusing to families. They're quite broad and non-descriptive. But Charlie needs help with everyday tasks like eating and dressing. He's non-verbal, he's not toilet trained, and he needs a routine that is easily frustrated. And like many people in his situation, Charlie has had a multitude of different genetic tests, but still remains without an answer. And this was the quote from his father, James, about what that meant for them. So without a diagnosis, we had no idea of his prognosis. And so for the next 11 years, we lived in the dark, raising a child with no handbook, feeling alone and blaming ourselves. And this is a kind of common feeling among these patients, these families with undiagnosed children. So developmental disorders like Charlie's are actually pretty common. So around 1 in every 200 children are born with a severe developmental disorder. So that corresponds to about 3,500 per year in the UK, or around 700,000 per year across the world. But the majority of those children do not have a genetic diagnosis. That means that we cannot find, despite suspecting that for the majority of these individuals, the cause is genetic, only about 40% of them can we actually pinpoint what that genetic cause is. And this is really difficult. Why is it so difficult? Well, we each have a lot of DNA in each of our cells, about 3 billion what we call base pairs, which is actually described as a series of letters of A's, C's, T's, and G's, or here I'm in RNA actually, so U's rather than T's. But out of those 3 billion base pairs, often it's a single change in that letter code. So here, a G to a C, that can be enough to cause a disorder as severe as Charlie's. So actually computationally trying to find that single letter change amongst 3 billion base pairs is a really hard task, akin to the old adage of trying to find a needle in a haystack. So in the clinical genetic sense, we often focus on genes within our genome. So we have about 20,000, 18 to 20,000 genes in our genome that code for proteins. And proteins are thought to be the workhorses that do most of the processes within our cells. So often we focus on these particular regions of the genome as a first place to look for a diagnosis. But actually there are many more genes across our genome, about 25,000 annotated in humans to date, that are not translated into a protein and actually function as what we call non-coding RNAs. So RNA is normally thought as the intermediate between DNA and protein, but actually for a lot of genes, it's the RNA that actually has a function in the cells. And as far as clinical diagnosis is concerned, the protein coding genes, the ones shown here in orange, have been really well studied. That's where we mostly look for diagnoses. Whereas the red genes on the right-hand side, these non-coding RNAs, are not very well understood and have not been searched for genetic diagnoses. And because of our prior that these protein coding genes are going to be where we find these diagnoses, actually when we sequence the DNA, so to look at that genetic code in an individual with a disorder like Charlie's, we actually often only look at the sequence of those protein coding genes through a process called exome sequencing. So here we're just, these teal or green genes here are protein coding genes. So we look at the DNA fragments of just those genes. And this the purple gene here, which is a non-coding RNA, we don't have any sequencing data for that gene. But more recently in the UK and elsewhere, we use a technique called genome sequencing, where we actually look at the DNA sequence across the entire genome, including those protein coding genes, the non-coding genes, and also all the regulatory sequence that comes between them. So why do we use exome sequencing? Why don't we always just look at all of the DNA? Well, exome sequencing is much quicker, it's cheaper, it's much easier to analyze because we're only looking at a small number of genes where we have a prior of what we expect a variant to be doing. And we only look at about 2% of the DNA. Basically just makes our lives a hell of a lot easier. Counter to that, if we use genome sequencing, it's very expensive. It generates a hell of a lot of data, but it allows us to look at all of the DNA, including those non-coding RNAs. And I'm going on about those non-coding RNAs for a good reason. So in the UK, many years ago now, our government actually invested in heavily in genome sequencing. And for individuals like Charlie, who were without a diagnosis, they were enrolled into the Genomics England 100,000 Genomes project, and genome sequencing was done normally on them and also their parents. So we can look for changes in the DNA sequence in those individuals and compare them to their parents to find changes that were different and might be causing their disease. I'm not going to talk about this in detail because the wonderful Susie Walker is going to talk more about Genomics England later. But just to highlight what we can do with this data, in the clinical genetics sense, we normally have a single patient that walks into a clinic and we look at their DNA sequence and try and find the cause of their disease. And that's really, really difficult when you've got about 1 million different DNA changes across their genome. But if we suddenly have a cohort of individuals, all who remain undiagnosed, and can find individuals with similar-ish phenotypes, we can suddenly find things in common or patterns in this sequence. So here I'm showing you that all of these individuals have the exact same DNA change shown here in pink, this gene. And then we can suddenly say, hang on a minute, maybe that is the cause of disease. And Yuan's going to talk about this in more detail up next, but this is how we came across the gene ReNU42, which encodes one of these non-coding RNAs as a cause of what is now called ReNU syndrome. So I'm going to very briefly talk about a few highlights. So ReNU42 is a really, really tiny gene. I told you there's 3 billion base pairs of DNA. It's only 141 nucleotides long, or base pairs long. So it's really, really short. And actually all of the variants or the DNA changes that we find in patients are clustered in a really small part of it. So 18 base pairs of DNA. And the vast majority of individuals have actually the same specific change to that DNA, which is an insertion of a T in the DNA or a U into that RNA, which is found in about 3/4 of all individuals. And it was this highly prevalent variant that meant that we could identify this gene in the first place. And as I said, Yuan's going to talk about that in more detail. So why is this important? Why is this finding so remarkable? Why are we having this event today? Well, it's estimated that around 100,000 individuals worldwide have ReNU syndrome. So they were all individuals that previously did not have a diagnosis. But what makes ReNU syndrome particularly special is it's one of very few diseases that have been linked to one of these non-coding RNA genes. And the ones that have been identified already are known to cause disease in maybe a handful of patients, nowhere near 100,000. So, and the other thing here is that we did not expect to find a new disease that explained this many undiagnosed patients. The majority of diseases that are this common were actually discovered a decade or more ago. So this was basically just hiding in a part of the genome that we just didn't look in. And it's now kind of blown open the rare disease field to say, actually, we need to pay attention to these non-coding RNAs. And I said that all of those variants are found in this really, really tiny region of 18 base pairs. And this in itself is just a kind of really confusing bit of biology because we don't really know why. So these variants are all what we call de novo changes. They exist in a child, but are not found in either of their parents. They've newly arisen in that child. And we expect de novo variants to be somewhat random occurrences. They seem to be somewhat randomly spaced across the genome. But to find so many individuals caused by variants in the same small region, with the majority of them having the exact same de novo change, is really, really surprising. And we're still trying to dig into why on earth that is the case. But what it does allow us to do is to get a diagnosis back to families like the wonderful Charlie. And Sarah's going to talk to you a lot more about the impact that this discovery has on patients. But just to pull out 2 quotes from a wonderful article in the Sunday Times that Charlie's dad, James, wrote. But firstly, a lot of mothers think that if their child has a severe developmental condition, it's something they did. Maybe it's that 1 glass of wine they drank during pregnancy, and that's the reason their child has all of these issues. So to be able to give them a diagnosis that tells them that it's not their fault is a huge, huge relief. But also it gives them a community. So James's first quote was that they're raising a child without a handbook, with no prognosis, no idea of what the future looks like. But you can suddenly give these families a community where they can see other children like their child, maybe that are 10 years ahead, and know what the future looks like. And that's really, really powerful. So this is a map created by the wonderful families, which shows you all of the individuals that have been diagnosed with ReNU syndrome and are part of the wonderful Facebook group that was created by a mom called Jessica. This map fills me with both joy and also horror. So joy in the fact that there are so many dots on this map, when actually we first put the preprint out about this in April last year. So over a year, we've made this many diagnoses. But the horror is in that it just shows us the disparity of access to things like genome sequencing. So genome sequencing, as I said at the start, is really expensive. So it's a luxury of wealthier nations. And you can see that these diagnoses cluster in Europe, Australia, and the US. So we've got a lot of work to do to try and get those diagnoses to patients actually around the world. But this discovery has had a broad impact. So as I said, there's hundreds of patients have been diagnosed to date. The gene is now included on diagnostic panels around the world. So if an individual presents to clinic and gets genetic testing now, they should be given this diagnosis immediately and not have the long diagnostic odyssey that is common in a lot of patients with developmental disorders. There is an absolutely incredible patient organization that has already been formed. They've got charity status in the US, are going for that in the UK. And they are amazing and really, really driven. And they're actually having their first meeting in New York in July. What this discovery also allows us to do is for all of these individuals to investigate whether we can create a therapy. So we're at an amazing time now for genome-targeted therapies. And there is actually a hope that we might be able to find a treatment that can help some of these individuals. And Stefan's going to talk more about that later. And finally, this is made a bit of a shift in rare disease research. So there was a lot of debates as to whether we could gain more value by looking at genomes over exomes, with a lot of people still thinking that all of the common disorders would be caused by protein coding genes. So what we've done is made people to sit up and pay attention to the fact that we need to also look in these non-coding RNAs and elsewhere in our genome. So I just want to touch before I finish on the name. So how did we get to ReNU syndrome? Now, I learned a lot about syndrome naming when we came to this discovery. You can't just call something after a gene. You can't call something ReNU42 syndrome, because what happens if you suddenly find two disorders caused by the same gene? Maybe one is dominant, so caused by a variant in a single allele, and one is recessive, so you need to impact both alleles. They're not going to be the same syndrome, so calling something ReNU42 syndrome would be very confusing. We also, there's a thing called OMIN that actually names a lot of these disorders, and they proposed a name that was NEDHAFA. I have no idea what it stands for. It's basically an acronym of symptoms, which the families obviously absolutely hate. It's really negative. It's about the issues that these children face. And one of the things in that crazy acronym is absent language. And actually, while a lot of these children are nonverbal, some of them can speak. Some of them can even speak fluent language. So they actually don't feel like they always belong to that syndrome name. So what's the alternative? This is the reason we end up with things like Parkinson's or Alzheimer's, or things that are named after people, because that's a better alternative than either a gene name or a symptom name. So instead, we use this wonderful quote that was given by Nicole Cedar, who's shown here with her daughter Mia Joy, where she said that they like to refer to ReNU42 as ReNU, which is a really clever play on that gene name of ReNU for ReNU, as their family is being renewed by this new information and hope for the future. So that's where we came to ReNU syndrome. It's official, it's in OMIM. There we go, there's the wonderful acronym that NEDHAFA stands for. But yeah, so we managed to, with the families, convince OMIM they should change this name, and it is now ReNU syndrome. So with that, I'm just going to leave you with some of the wonderful faces of individuals with ReNU syndrome and pass over to the wonderful Yi Yang, who actually made this discovery in the first place.